Phase 3, Adaptive Immune Response. The benefits of an adaptive immune response are 1. Specificity. It's the ability to specifically recognize and mount a response against almost any pathogen. Antigens are recognized by receptors on the surface of B and T lymphocytes. 2 is immunological memory. The first exposure to a pathogen is called a primary adaptive response. Symptoms of a first infection, called primary disease, are always relatively severe because it takes time for an initial adaptive immune response to a pathogen to become effective. Upon re-exposure to the same pathogen, a secondary adaptive immune response is generated, which is stronger and faster than the primary response, often eliminating the pathogen before it can cause damage or even symptoms. This secondary response is the basis of immunological memory, which gives us immunity. Self-recognition, the ability to distinguish between self-antigens, those that are normally present in the body, and foreign antigens, those that might be on a potential pathogen. As T and B cells mature, there are mechanisms in place that prevent them from recognizing self-antigen and preventing a damaging immune response against the body. When these mechanisms fail, their breakdown leads to autoimmune diseases. Lymphocytes, B cells, T cells, and plasma cells. As stated above, lymphocytes are the primary cells of adaptive immune responses. The plasma cell, or a B cell lymphocyte that has been activated through exposure to an antigen and produces antibodies against that antigen. There are 5 classes of antibodies: IgM, IgG, IgE, IgA, and IgD, each functioning in different ways. IgM promotes chemotaxis, opsonization, and cell lysis, making it very effective antibody against bacteria in early stages of a primary antibody response. IgG is the one that crosses the placenta to protect the developing fetus from disease and exits the blood to the interstitial fluid to fight extracellular pathogens. IgA is the only antibody to leave the interior of the body to protect body surfaces. IgA is also of importance to newborns because this antibody is present in mother's breast milk, also known as colostrum, which serves to protect the infant. IgE is associated with allergies and anaphylaxis. The next type of cell is a T cell. Different T cell types have the ability to either secrete soluble factors that communicate with other cells of the adaptive immune response, or destroy cells infected with intracellular pathogen. Cytotoxic T cells kill target cells by inducing apoptosis using the same mechanism as natural killer cells. Killing a virally infected cell before the virus can complete its replication cycle results in the production of no infectious particles. The next type of cell is a helper T cell, or Th cell. This cell releases cytokines, which help to develop and regulate other immune system cells. Suppressor T cell, also called regulatory T cell, control T cell response in order to prevent too many T cells from being formed during an immune response. Memory cells. B cells and T cells form during primary exposure to a pathogen, remain in the body for a long time after an infection, and are able to mount a fast and effective immune response to a pathogen if it is encountered a second time, preventing the pathogen from causing disease. Active versus passive immunity. Immunity to pathogens and the ability to control pathogen growth so that damage to the tissues of the body is limited can be acquired by 1. The active development of an immune response in the infected individual, or the passive transfer of immune components from an immune individual to a non-immune one. The downside to this passive immunity is the lack of the development of immunological memory. Once the antibodies are transferred, they are effective for only a limited time before they degrade. So an active immunity is a resistance to pathogens acquired during an adaptive immune response, the result of memory cells formed during the adaptive immune response of a pathogen. Through a vaccine response is through vaccination, one avoids the disease that results from the first exposure to the pathogen, yet reaps the benefits of protection from immunological memory. Vaccination was one of the major medical advances of the 20th century and led to the eradication of smallpox and the control of many infectious diseases, including polio, measles, and whooping cough. With passive immunity involves the transfer of antibodies through an immune person to a non-immune person. An example of this is transplacental antibodies from mother to fetus, and maternal antibodies in breast milk protect newborns from infections. Immunoglobulin injections taken from animals previously exposed to a specific pathogen is a fast-acting method of temporarily protecting an individual who was possibly exposed to a pathogen. Evasion of the immune system by pathogens. The immune system and pathogens are in a slow evolutionary race to see who stays on top. Early childhood is a time when the body develops much of its immunological memory that protects it from diseases in adults. Pathogens have shown the ability, however, to evade the body's immune responses as described below. Protective adaptations. It is important to keep in mind that although the immune system has evolved to be able to control many pathogens, pathogens themselves have evolved ways to evade the immune response. An example is the Mycobacterium tuberculosis, which has evolved a complex cell wall that is resistant to the digestive enzymes of the macrophages that ingest them, and thus persist in the host, causing the chronic disease tuberculosis. Multiple strains. Bacteria sometimes evade immune responses because they exist in multiple strains, each having different surface antigens and requiring individual adaptive immune responses. One example is a small group of strains of Staph aureus called methicillin-resistant Staphylococcus aureus, otherwise known as MRSA, which has become resistant to multiple antibiotics. Antigen mutation. Because viruses' surface molecules mutate continuously, viruses like influenza change enough each year that the flu vaccine for 1 year may not protect against the flu common to the next. New vaccine formulations must be derived for each flu season. Genetic recombination. An example is the influenza virus, which contains gene segments that can recombine when two different viruses infect the same cell. Recombination between human and pig influenza viruses led to the 2010 H1N1 swine flu outbreak. Immunosuppression. Pathogens, especially viruses, can produce immunosuppressive mo- molecules. molecules that impair immune function. Tissue transplantation. With the use of tissue typing and anti-rejection drugs, transplantation of organs and the control of the anti-transplant immune response have made huge strides in the past 50 years. Immunosuppressive drugs such as cyclosporine A have made transplants more successful, but tissue matching is still key. Family members, since they share a similar genetic background, are much more likely to share MHC molecules than unrelated individuals do. One disease of transplantation occurs with bone marrow transplants, which are used to treat various diseases including SCID, SCID, and leukemia. Because the bone marrow cells being transplanted contain lymphocytes capable of mounting an immune response, and because the recipient's immune response has been destroyed before receiving the transplant, the donor cells may attack the recipient tissues, causing graft versus host disease. Symptoms of this disease, which usually cause a rash and damage to the liver and mucosa, are variable, and attempts have been made to moderate the disease by first removing mature T cells from the donor bone marrow before transplanting it. Immune responses against cancer. It is clear that with some cancers, like Kaposi's sarcoma, for example, that a healthy immune system does a good job at controlling them. This disease, which is caused by the human herpes virus, is almost never observed in individuals with strong immune systems. Other examples of cancers caused by viruses include liver cancer caused by the hepatitis B virus and cervical cancer caused by the HPV or human papillomavirus. As these last two viruses have vaccines available for them, getting vaccinated can help prevent these two types of cancer by stimulating the immune response. On the other hand, as cancer cells are often able to divide and mutate rapidly, they may escape the immune response, just as certain pathogens such as HIV do. There are 3 stages in the immune response to many cancers. 1. Elimination occurs when the immune response first develops toward tumor-specific antigens specific to the cancer and actively kills most cancer cells. Equilibrium is the period that follows, during which the remaining cancer cells are held in check. Escape of the immune response and resulting disease occurs because many cancers mutate and no longer express any specific antigens for the immune system to respond to. This fact has led to extensive research in trying to develop ways to enhance an early immune response to completely eliminate the early cancer and thus prevent a later escape.